COVID-19 has forced many of us to accept a new normal. This includes staying home, but remember, you don't have to feel trapped at home. Get outside, get some exercise, play with your pets, even though mine's being lazy, and even take them on a car ride. But remember, stay 6 feet away from strangers if you go out anywhere.
This is a video depicting the interaction of a SARS spike protein receptor binding domain and a neutralizing antibody. Its PBD code is 2GHW. As you can see, the molecule has many nitrogen and oxygen containing amino acid residues with carbon chains. The protein spike itself and the antibody are both considered glycoproteins, which are a type of carbohydrate derivative. These are proteins that are covalently bound to oligosaccharides, which can contain anywhere from 1 to 30 plus carbons. The carbon chains are either O-linked to a serine or a threonine amino acid residue, or they are N-linked to an asparagine residue. This could help explain why there are so many oxygen and nitrogen atoms found in the complex. The antibody, according to the UN et al. Article, does not bind to the SARS spike protein receptor active site. This means that it acts as an allosteric regulator to the virus. This means that the antibody binds to some other location on the protein spike and inhibits or activates the virus. Allosteric regulation is a regular part of metabolism and is often occurred between different biological molecules. In this case, the antibody is trying to inhibit the infectious nature of the virus. This antibody is comprised of a light chain and a heavy chain and was obtained from a survivor of SARS. Although SARS does not bind to the SARS-CoV-2 RBD, it does not bind tightly enough to prevent the cellular infection. Steric hindrance is yet another issue in the binding of SARS to the current COVID-19 RBD. The RBD and SARS only interact when the RBD is in the up or open conformation, as in the down or closed conformation. The SARS variable domain would conflict with the S2 under the RBD in the open formation. However, steric hindrance still occurs.
Masks are a great way to help protect yourself against COVID-19. Although medical masks should be saved for frontline healthcare workers, homemade masks are a good alternative. Homemade masks should be multiple layers thick and washable, and should also fit snugly yet comfortably against your face and be secured with ear loops or ties. Although masks are good for helping to protect yourself, they can't completely protect you, so you should avoid touching your eyes, nose, and mouth. Together we can help stop the spread of COVID-19.
The protein spike of COVID-19 is comprised of homotrimers, which extend outward from the surface of the virus. The protein spike has two subunits that aid in host cell infection. The S1 subunit facilitates binding to the receptor of the host cell, and the S2 participates in the fusion process between the cell and the viral membranes. The glycoprotein spike is further altered by proteases supplied by the host cell, which cleave the protein at the S2 site, initializing the fusion process. The S1 subunit has two conformations, open and closed, which can be determined by looking at the V-shaped point on the protein. In the closed form, the HACE2 recognition motifs are inaccessible, as they are underneath the promoter interface. In the open conformation, the protein can interact with ACE2 at the surface of the host cell, allowing it to gain entry into the cell through the cleavage fusion processes. ACE2, or angiotensin converting enzyme 2, is the host cell receptor that S1 subunit of the COVID-19 spike protein binds to. ACE2 can be found in various locations of the body, including the heart, intestines, kidneys, lungs. ACE2 proteins are considered type 1 membrane proteins. In a full ACE2, a claw-shaped N-terminal PD and C-terminal collectrin-like domain can be observed.
Thank you!
Go.
Even though the coronavirus may keep us inside, it's still easy to keep in contact with our family using FaceTime. Say hi, Mom! Hello!
Remdesivir is a potential drug to combat the spread of COVID-19. But before we talk about how remdesivir works, first we have to talk about how NTP usually works. In RNA polymerase, the aspartic acid residues in the core coordinate metal ions, which coordinate and stabilize the phosphate groups of the NTP. The RNA transcript's last base forms a phosphodiester bond with the NTP. This bumps the electrons from the oxygen-phosphorus bond onto the PPI. This mechanism ends with the formation of an RNA strand and the new nucleotide attached. Remdesivir binds the same way in the core of RNA polymerase. Remdesivir binds in the core of the RNA polymerase and it acts as a competitive inhibitor. It gets transferred into this analog, which has two slight structural differences between the RDV-TP and the NTP. The remdesivir gets incorporated into the mRNA sequence, and as a result, translation is cut short whenever they appear. For example, when AUG binds to the tRNA in the ribosome, methionine is added to the amino acid sequence. When remdesivir NTP is added to the codon, it does not get coded and translation stops immediately. This results in fragments of polypeptide sequences that are not useful to the production of the virus. Here's a quick overview of how competitive inhibition takes place. The enzyme usually binds to NTP, which produces an RNA strand and the enzyme, obviously, but sometimes it can also bind to RDVTP, which produces an unusable strand. It ends up inhibiting the viral protein process. So COVID-19 would end up with less viral proteins to work with.
This image represents the coronavirus's papen-like protease. This protease is essential for the replication of the coronavirus once in the cell. Antiviral medication developments are trying to target this protease so replication can be stopped within the host and its cell. These targeted drugs can stop the replication as well as cause cell death surrounding infected cells, so these drugs can inhibit the spread in two different ways at the same time: by cell death of surrounding healthy cells and the inhibition of cell replication. Soap acts as a superfactant that has a water-loving side and a water-hating side, kind of like the phospholipid bilayer within the cell membrane. Hydrophobic and hydrophilic. The hydrophobic sections grab onto the oil residues that are found on your hand that you pick up from germs and other surfaces. The hydrophilic section likes to cling onto the water. When you wash your hands, the soap, the hydrophobic side, likes to grab the oils. When you rinse your hands with water, the hydrophilic side rinses away the dirt and holds on to the oils, cleaning your hands. Current recommendations for washing hands say that 20 seconds is good enough, but I say sing the happy birthday song 2 times.